I would like to welcome everybody on this presentation of recognizing magnification levels in microscopic snapshots. Before we start our presentation, I would like to thank IEEE for hosting 40-second virtual conference of engineering in medicine and biology. A short introduction about myself. I am Manit Zaveri from University of Waterloo. I am pursuing my master's in applied science from the Department of Systems Design Engineering. Apart from that, I am a graduate research assistant at Kimia lab. The main goal of our research lab is to develop an image search engine specifically for the role of pathology diagnosis. When we talk about histopathology, the common consensus or the common diagnosis is being conducted by a biopsy sample from the patient being observed under the microscope, and a detailed report is being prepared by a pathologist regarding the specimen and its characteristics. Hence, we can say that microscopic analysis is the gold standard in pathology. When talking about digital pathology, we mainly have two modalities which are responsible for giving us digital images. First is light microscopy or photomicroscopy. Light microscopy attached with camera is responsible for capturing snapshots, while we have whole slide scanners, which are emerging technology. Whole slide scanners have far more resolution and image quality as compared to simple camera attached light microscopes. But apart from that, microscopy is far more popular than whole slide scanners because of its prevalence since centuries. Whole slide scanners are developed in recent decades as compared to microscopes since centuries. The images coming from microscopes are low quality images. As a result, they are static images at specific magnification in which a pathologist have conducted the diagnosis of a particular glass slide. While for whole slide images, we have access to all the different magnifications because of their file format in which the whole slide images are stored. Hence, we can see a tremendous amount of difference between two modalities. But what about the data set being generated camera, from the camera attached to light microscopes? Should we just let them pass on with history, or should we use them? Of course, we will use them. So we are trying to classify the magnification of those camera captured snapshots. So because of the WSI technology being new and emerging technology, it is not very popular as compared to microscopes. As a result, most of the pathologists tend to use microscopes while we are talking. We try to generate a diverse data set which is composed of multi-magnifications, which will be further used for training an entire CNN from scratch. So instead of using pre-trained networks, we will be able to use pathology trained network. For this particular project, the dataset that we used composes of 700,000 images, specifically coming from 33 different organs of human body. We have 5 magnification levels of 2.5x, 5x, 10x, 20x, and 40x. So the 2.5x captures the maximum field of view with less attention to the minute details, while 40x is the maximum magnification in which we see the most enlarged tissues from a particular glass slide. A simple topology of how the magnification works is being shown in figure 2. So we try to deploy handcrafted features and deep features so we can conclude which features are best for magnification classification. As a result, we deploy local binary patterns, which are so far the best handcrafted features, and DenseNet 121, which is pre-trained with natural ImageNet weights. The classifiers that we used for this project are random forest and shallow classifier. As a result, we can say that we are fine-tuning the DenseNet But for this project particularly, we are not touching the architecture of the dense net. As a result, we will keep a shallow classifier instead of fine-tuning. The evaluation of the network, the evaluation of entire framework is done using two metrics. First is the overall accuracy, and the second one is the individual class accuracy score, which is focusing on the misclassifications at each magnification level. An entire framework is being seen in this diagram, and we see that the image patches are being pre-processed. By pre-processing, we normalize the mean and standard deviation, and we pass the images without changing the weights of the network to extract the features. Using this, we get an image shaped 800 pixels by 800 pixels into a feature vector representing an array of length 1024, that is 1024. These features are used to train a classifier. While we use only 80% of the data to train the classifier and use 20% to test the classifier model. This is done over the fold of 5 times to validate the model accuracy. When we change the feature extractors from DenseNet to local binary patterns, the length of features respectively changes for 10, 12, and 24. So an image of 800 pixels by 800 pixels is being represented with only a feature vector of length 10. Coming to the results, here are all the results across all the folds and all the feature extractor and classifier combinations. We can see that the best accuracy is obtained by deep features when combined with shallow classifiers. While for the local binary patterns, the best accuracy score that we obtained is 63.3% with a deviation of 0.5%. In past, we have seen that local binary patterns have outperformed deep features, but for the task of magnification classification, we see that deep features are far more better than traditional handcrafted features. While comparing about the features, we also compare the accuracy of the classifier. The random forest classifier obtains 90% accuracy when trained with deep features. Coming on the second type of metric, we analyze the misclassifications at each magnification level. So for these shallow classifiers, we see that the maximum misclassifications occurred at 5x magnification. Well, that may be because of the small or the confusing area of field of view of images. Well, it cannot generalize, the network cannot generalize 5x images as much as other magnification levels. While we see that most of the 5x magnification levels are misclassified in 2.5x and 10x, which is adjacent to the magnification level. Well, that makes some sense because most of the content in 2.5x is being overlapped in 5x. As a result, it becomes evident to get misclassified in the adjacent magnification levels. While for the random forest, we see that the actual correct classification for 5x stands only at 82%. As a result, most of the misclassifications for random forest are even in the adjacent classes. So that was the actual framework of how the experiments were conducted. Coming on the future scopes, we aim to train a multi-magnification neural network from scratch using a balanced dataset created using the proposed framework. This will subsequently improve the image search results, and if we are using some other new dataset being developed for pathology, we can use the trained network to extract features which will be of high quality as compared to ImageNet weights. This use of huge data repositories will bring a variation in staining modalities, tissue origin, and magnifications, subsequently elevating the performance of all machine learning applications. I would like to thank everybody for watching this presentation. And for more questions, I would like to welcome you on our webpage, www.kimialab.uwaterloo.ca/Kimia. Thank you.